And for the other cell line, for the colorectal cell line, we see that we see a different pattern here, and I think I made a mistake. I think that's right. Yes. Yeah, actually I missed a slide here in my new version. So it looks like that. To summarize the results, we would say that both types of DDWs, A and B, showed no apparent effects on proliferation of T47 and HT29 cell line. Anti-proliferative effect of doxorubicin was slightly increased when prepared in DDWs in comparison to doxorubicin alone. The S phase in cell cycle pattern of T47 cells was significantly increased only in doxorubicin prepared in type A of DDW. In HT29 cells, the G2M phase of cell cycle was significantly increased in both DOX-A and DOX prepared with DDWB in comparison to doxorubicin alone. The T47 cells exposed to DOX-A and DOX-B in comparison to doxorubicin alone showed increased apoptosis and necrosis, respectively. In the HT29 cell lines, the G2M phase of cell cycle was significantly increased in both doxorubicin A and doxorubicin B in comparison to doxorubicin alone. To conclude, our data supports the previously reported findings on significant potentials of deuterium depleted water on cancer cells to enhance the effectiveness of chemotherapy. And I thank you very much for your attention, and if you have further questions or you need further explanation, you could contact Dr. Azizi at his email address. Thank you very much.
So the talk is open for questions. I'd just like to ask you one question. It's nice to keep the investigators blind. So the talk is open for questions. I'd just like to ask you one question. It's nice to keep the investigators blind, but the audience is still blind. Do you know what the ppm concentration in A or B was now?
Yes, actually, I've got them here. The A was 50 ppm, and the other one was 105 ppm.
So A was 50, and
A was 50 ppm,
B was
and
105.
the B was 105 ppm.
It seems it was a triple-blinded study.
It was a blind, actually, yeah, it was. It was.
So, any questions, though? Go ahead.
Do you have any, let's say, idea on the mechanism of doxorubicin which might be enhanced by the presence of...
Actually, I personally don't have any idea. I think
Oh.
You better ask Dr. Azizi.
Right. Okay.
I don't know. Really.
Actually, doxorubicin is, I believe they used it as an antibiotics first, and then it had anti-proliferative effects, and by default, it seems that it, as far as we know, because we did profile doxorubicin with some other anti-cancer agents, it increases the oxidative branch of the pentose cycle. So what I would assume there's a relative compensation on cells adapt to transcurase inhibition by upregulating the oxy branch, and if it's consistent with DDW somehow decreasing or inhibiting this proton exchange in the glucose 6-phosphate dehydrogenase reaction, that can be one mechanism. But this is just a contemplation, and Dr. Usisi might be able to answer it best, but this would be my take on any mechanism that is involved.
I would like to some short note, because I am thinking, so finally you repeated that checking the cell growth after 24 hours, you couldn't confirm that the deuterium depleted water itself can inhibit the cell growth. And one question is, which could be common with your experiment, the time frame, that it's 2 days or even longer. But the other question came to my mind that when you put the cells into the media and put it in the chamber, keeping on 37 Celsius with a 100% humidity, do you try to avoid the exchange between the media and between the humidity? Because if you don't,
no, we didn't.
Okay,
No.
if you don't do that, finally, gradually the deuterium concentration will increase in the media, and you are not testing a media with low deuterium level. And I don't know whether you try to avoid it. Not close, because there is an exchange, because the pH is regulated and the carbon dioxide is going and the water. So that could be, I think, a disturbing factor. Okay, just came to my mind, because I know I was sure I also tried to avoid any exchange between the air. So that can be a disturbing. Okay, thank you.
Okay, thank you.
All right, so thank you, Dr. Hussein. So